welcome back. As you could see, our 15 minutes commercial break became almost half an hour. So I have to keep the introduction little brief, but today's first Foundation Day lecture, there will be two Foundation Day lecture as it's been already announced. And the first lecture will be given by very distinguished personality, a renowned scholar, and at the same time, and very efficient administrator, Professor Suman Chakraborty, Director and Professor at the Department of Chemical Engineering of Mechanical Engineering, Mechanical Engineering of IIT KGP. We are honored to have him with us. Professor Chakraborty's research interest lies in fundamentals of macro and nanoscale fluid dynamics, miniaturization, and its applications focused towards sustainable technology goals for promoting good health. He is the inventor of paper and pencil microfluidics technology, a new class of electrically manipulative miniaturized devices that does not require any sophisticated fabrication facility. Professor Chakraborty is also known for developing disruptive medical diagnosis technologies for the underserved. His work has been recognized internationally and nationally through numerous awards and fellowships of prestigious societies. I'll mention only a few just for the sake of time constraint. Professor Chakraborty has been awarded the Shanti Sarup Bhatnagar Prize. He has been elected as Fellows of the American Physical Society. In fact, this is the second engineer after Ashutosh Sharma I know of being Fellow of American Physical Society, Royal Society of Chemistry, American Society of Mechanical Engineers, and all the national academies of the science and engineering. He is recipient of Infosys Prize, GD Birla Award for Scientific Research. I must stop here, otherwise I will eat up even the time of this lecture. So I welcome Professor Chakraborty to deliver this lecture.
Good afternoon, and thanks Professor Tanushree Shah for inviting me in this very auspicious occasion where the great institute is completing 40 years. And I'm also very much honored and humbled to be there in presence of very distinguished luminaries like Professor Yadav and Professor Varup Rai Chaudhuri, Professor Shomit Roy, many others who are like my teachers. And therefore, it is a great opportunity. In fact, those who have been in IISc are literally teachers. So, it is a great honor for me to be there in this platform and sharing my views on something which I feel is very important, no matter whether one is doing basic science or so-called applied science. That the main theme that I would like to emphasize on is that we all, at least those of us who are from engineering background, we all emphasize a lot in doing work for the practical purpose. At the same time, the fundamental origin of our work is of no difference as compared to what a scientist will do. It's a lab. Now, where the journey shapes up from there to the outcome is something which is very decisive, and I would like to connect that with the living world. Now, all of us have gone through transformational experiences in education and research, and the ones who are very senior here, they have seen many such sort of changes. When we were students, of course, there was no question of AI being dominating anything. But when some of our teachers were students, they may not have seen computer in their student life. So things have changed significantly. But for a long period of time, we were having a philosophy of what we say as discipline-oriented education. And to a large extent, it has served the purpose and it is going to serve the purpose. But if we look into the modern era perspective, and typically the manner in which different disciplines have come very close to each other and they work among themselves, the disciplinary silo is not really turning out to be very effective. But at the same time, the fallacy is that while we are having many centers, many research units, labs, etc., we are still sticking to the sanctity of this disciplinary silo. Not only that, people who are not sticking to that are getting penalized in some way or the other. I still find in IIT Kharagpur, also in some departments, they will not shortlist a person or faculty if the person doesn't have a degree in that in the undergraduate or masters. So on one side we talk about great things interdisciplinary, but on another side we are still very much comfortable in having the strong disciplinary prominence, and this contradiction needs to be shifted. So I particularly put this up because I know that this is a basic science institution, and although there is a lot of work that goes on which is actually no less than what an engineer will do, but it is tagged as a basic science. But over my entire learning, I always struggle to define a problem. Is it a physics problem or an engineering problem? In many cases. And at the end, I realized that it is neither physics nor engineering or whatever. It's a systems problem which is connected by many factors. So, in my own journey in studying the foundations of fluid mechanics to micro nano systems and biomedical interfaces, I have myself witnessed this shift, and not that I was always ready to accept this shift, but this is what you can summarize as scientifically the perspective change from equations describing some idealized flows to modeling of blood flow in living microvasculature. Both may be theoretical or simulation work, but there is a huge difference. From microchannels in silicon to point of care diagnostic platforms serving rural communities, which are not definitely on such classical platforms, and from controlled lab validation to deployment in climate vulnerable regions, where any finding in the lab is confronted with significant challenge. So what I have realized, and I wish I would have realized much earlier in my student life, I always thought that lab is the end point. And once you do something good in the lab, you publish a paper, and that's the greatest thing. Now I realize, at least in the kind of work that I am engaged in, that the lab is the beginning, and there are lots of things that can happen beyond that. So then, one of the learnings that I learned out of hard experience is that while naturally we have a tendency of what I call as reductionism, but to create an impact and to solve a real life problem, maybe as a problem solving or in a mission mode, we ultimately would need convergence of many things. And this convergence of many things, how they are orchestrated carefully, that actually leads to the success story of the problem that is being solved. Now, if I could in a very simple way have a chance of telling that how science in the living world appears to be somewhat striking as compared to the science in the idealized world. The living world is uncertain, non-linear, ethically charged, and it resists simplification. In fact, on a lighter anecdote, I have a knack of sort of preparing talks for primary school maths in what you say as Bangla language, regional language. And this knack I developed when I tried to teach my son about those things. And then I realized a lot of gaps in actually our primary level school education, which has given rise to such oversimplified thoughts. So if 5 people working 10 hours per day does a job in say 20 days, then 30 people working 1 hour a day, how much time the person will take? So we will use unitary method, which is a linearization. But a large group and a small group, and number of hours a day more or less. So at that level, we would never try to imagine that if the person works more than 6 or 8 hours a day, overtime will come. And beyond that, if large number of people are forced to work for beyond the critical hour, then labor union will strike and work will stop. So in that way, whatever simplified linear things that we learn, there is a significant difference. So in fluid mechanics, even in the basic perspective, people were once satisfied in predicting sort of very simple laminar flows between the parallel plates or pipes, etc. But today we are studying deformable biological tissues coupled with electric field, molecular interactions, and data-driven prediction. And the property of that tissue is not like a rubber or a steel. It is not known. And it comes from many different convoluted things, and some of those may be related to diseased conditions. In material science, which is of course such an overarching branch that it connects everything, we no longer ask, is it strong? Why I am telling this is because as a mechanical engineering student, we have been having a subject as strength of materials. And for that period, at least in our student life, I believe that strength of material is the ultimate thing. And now sustainability, circular, scalable, equitable, so many factors are coming. So that strength is of course important, but it doesn't tell the final story at all. In biomedical engineering, which I am going to focus on for some of the examples, a device is not successful just because it works in a lab. It succeeds only when it survives cost constraints, field variability, regulatory realities, and societal acceptance. And this is why I am trying to highlight on this, that this is where India science has great opportunity. For a long period of time, India science has done great work, and at least over the past few decades, outstanding publications or the fame of Indian scientists all across the world. This has happened. But if we ask ourselves with a real conscience that most of the times, what exactly we are satisfied with, in the name of cutting edge research, we are mostly satisfied by being a follower of something where a great breakthrough has happened somewhere. Be it nano, be it bio, or anywhere. And the reason why we cannot lead those is because of our limitation in resources. So although we have in principle the ability to go beyond that, but the greatest of the researchers in those areas have flown down from the labs which have such great instrumentation and facilities, that going beyond that and do another pioneering strike is not that difficult, but doing something incremental may be possible, and that itself gives you sure shot publications and all. People are usually sort of content with that. Now these kinds of India-centric problems where you have extreme resource poor settings and you need to have a technology which has to work there by whatever, that may be a black box. So that is a kind of a hard problem in public health technology, which unfortunately cannot be studied directly by even of the most resourced labs in the world, because they do not have the practical living field condition where they can reciprocate and test and develop their science background and the technology. And in fact, this kind of thing is so emphatic. Just my recent experience. So this year we got a few expressions of interest from MIT pre-med students to do internship in the medical technology at IIT Kharagpur. And it was a heavily competitive thing from the MIT student side. So now if someone just out of common sense would think that why those MIT students, MIT pre-med students are brilliant students. So why are they coming to IIT Kharagpur for their this internship immersion? If we deep dive a little bit, there are always factors like societal and cultural immersion. These are all very good, food, society and all those things. But only for that they will not come in a program. They might come for an excursion. So the kinds of the primary healthcare setups and technology being deployed in those setups, they actually want to develop a first-hand experience on that they cannot have in the MIT's lab. So this gives us, in fact, this is very what I would say interesting, because we have our inherent challenges which come from the diversity, the demography, the huge population, but this challenge is actually giving us opportunity to come up with scientific and technology breakthroughs. So from proof of concept to proof of impact, and from publication metrics to societal metrics, and from disciplinary depth alone to convergent intelligence, with AI coming in hand with some of these technologies. I think these are some of the pointers which are going to take whatever research journey that the great Indian scientists have done over the past few years. Now the fruits have to bear by keeping these facts in mind. So this is a science, this is a cartoon of a science in a living world. I myself did not draw it. I used the Gemini AI help to draw it, but I gave a lot of input to this, so that at the end Gemini understands what I want to do, or what I want to really represent. So this actually, in a way, represents even my own journey. When I was a student in Professor Pradeep Datta's lab, that was like that CFD simulation I used to do and enjoy. And from there to when I see the smiling faces of the young children where we deploy our technologies, this is a sort of, I would say, a big transformation that can even happen to an individual. So, of course, in IIT Kharagpur we have an opportunity where we have a systemic way in which we can do it, because we have several units where medical technology is being studied as academic courses, research programs, etc. And we also have a connectivity of that with the primary healthcare units, which are sort of digital health units established mostly with the support of the government and the NGOs at different extreme remote locations. And while telemedicine is able to connect us with those quite easily and connect them with doctors, but there will always be a physical gap if we are just depending on communication and internet technology. So what we can do, or what we have rather done, I would like to again sort of sum up in terms of some examples. And these examples are just to illustrate that how when we start one thing, we do not really know that how it will lead to, a solution will lead to another problem, because the solution may be good but it is not satisfactory, and that limitation of one solution actually drives the solution of the other. So the first example, let me just maybe run the movie and then I can talk in parallel to save time. So this is a simple blood test which normally people would require that venipuncture and do the test. So instead of that, it is like you have a paper strip and you have this lancet needle. You take one drop of blood, and if you look into it carefully, this looks like simple, but it is not that simple. In a way that the spot where the blood is being sucked, that blood has to be metered with a particular volume, 20 microliter, 30 microliter, like that. But we did not keep a pipette here, simply because the rural boys or girls who are handling these technologies get uncomfortable with pipette. In sometimes we have to use it. But so what we did is, it is just a sucking by capillary action on the paper, and the microfluidic design itself puts the metered volume of blood on that spot. Then there is a small membrane filter which will filter out the red blood cells, and then the plasma gets naturally separated, and there is a micro channel by capillary action. The plasma will move to the reaction spot, and then there are reagents which are commonly lyophilized, that means powdered, and you can just put a solution to make them active, and they will react and there will be a color change. And typically we use colorimetric reactions. Even 5-10 years back, colorimetric reactions were not considered to be quantitatively good. But now with the AI enabled image processing, the colorimetric reactions are damn good actually to come up with good quality quantitative results at very low cost, because you do not require very high end camera, for such purposes. So essentially, for example, for blood sugar, it's just like a standard glucose oxidase peroxidase reaction. And at the end, whatever product is developed, you need to just derive a side product which has a prominent color. And that color prominence subjectively can tell you certain things, but to do it objectively, we actually take the image, and for that this black box, entire black box is there. So inside you have controlled optics and electronics, and in that way the microcontroller will control the illumination, and you have a LED lamp which will illuminate a spot, which is the spot where the paper strip reaction has taken place. And then there is commonly an inbuilt camera with the device itself, because it standardizes the camera, and then you take the image and analyze the image. And these are all calibrated, so the same sample you do testing in an automated biochemistry lab, for example, and then develop a correlation. But it's not a simple y as a function of x correlation, because you are extracting different features from the image and correlate those features with the outcome. So it's a machine learning instead of a simple mathematical correlation, but the spirit is very similar. Now, with this kind of technology, so these were some of our early technologies. Maybe I would say about 15 years back, we started with those, and then we realized that some of the things where we could improve upon this to make the test faster is that when the plasma is flowing or any body fluid is flowing from the dispensing point to the test point by the microchannel, if it is moving just by capillary action, then it is fundamentally a diffusive transport driven process, and it has its own limitations in terms of the diffusion time scale. Now, how you can overcome it? Of course, you can use field mediated effects. And the paper which is commonly used for such low cost diagnostic purposes, of course not the usual paper, but these are filter papers of certain grades. Usual paper also could be adapted, but it requires much more modification. Now, if you are trying to have that electric field acting on the body fluid in a paper, it is very much possible, just like we have electrokinetics in a glass, because the paper has cellulosic group, material which has carboxylate group, and that makes its zeta potential naturally negative. So if you have an electric field, it will move like electroosmosis that you have in a classical channel. But the practical issue is that for that you have to embed electrodes. I mean deposit electrodes. And electrode deposition is a known technology. Those who have been working in MEMS and all, they are all familiar. But the question is that can you have a simple frugal electrode deposition technology which will be commensurate with the cost factor that these kind of devices are going to demand. So a bit of crazy idea at that time came to my mind, which I shared with some of my students, that you can, can't we just sketch with pencil? You can see these black boxes. So simple HB pencil sketch. And the intuition was that the lead of the pencil, this has graphite. So this is a primitive way of graphite deposition. One could imagine. And then if you connect it with copper wire and then put a battery, and at that time when we did, the battery technology was not as advanced as it is now, but overall this is the scheme. We found that yes, it is working, and it is the capillary transport, if you just compare these graphs, almost twice as fast if you change from 0 to 50 volt bias. So in that way, electrokinetics can help, and it can be combined with the traditional paper microfluidics. And this at that time we coined as paper and pencil microfluidics. Of course, we did not have a great vision that what we will do, but at least it gives me a satisfaction to see that one subject that has emerged from our lab, and many people are using this and citing this also. Now, when it is scaled up, it is not that people are doing a cottage industry by sketching in pencil. So there is a graphite deposition automated system which is actually enabling it to be scaled up. Now, again, if you see every little bit of difference that I would like to highlight, it emerges from an unmet need from the previous one. Now, those kinds of tests that I have shown, they are simple colorimetric tests, but there are many tests who, even if you do with optics, they are more fluorescence dependent tests. So the wavelength, particularly the wavelength of excitation and emission that you want to study, are immensely critical, and if you can do that, it will be very precise. But again, the challenge is that you would require that spectroscopy or fluidimeter, which is there as a part of many of the labs, but we are imagining that these tests are done almost on a field condition, so where these kinds of lab things are not available. So by that time we started with this project, which is like, how can we detect a very low concentration of an inflammatory marker in the blood? Inflammatory markers, I mean, some people would call it cancer markers, but not necessarily they are specific to cancer, like CA125 or PSA. So these are all inflammatory markers. Typically, these are very common. Also, breast cancer inflammatory marker. There are so many inflammatory markers are there, but these are not definitive tests for cancer. So these can only enable one to screen effectively, and the challenge is, while they can be screened, but a minimum concentration of that is required to detect it. So you cannot normally detect like nanogram per milliliter or something like that. That's not, even in the most sophisticated technology, it is not possible. So we try to see what we can do. So this is what is known as lateral flow paper assay, which is, you can say, a light version of ELISA. So you are trying to have antigen antibody conjugation. So you have one section of the membrane where you have the antibodies embedded, and if there are these antigens which are inflammatory markers, they will combined with the antibody, an antigen antibody complex will move by capillary action and get tagged with another antibody. And generally, like for COVID testing, for example, this tagging would give rise to a surface plasmon resonance, and then you get that test line on the rapid antigen kit of COVID. Now, but that requires a threshold concentration. That is why those devices are not very sensitive. So only when you have very high load of the disease, then only you can capture. But here the purpose is different. So what we did is we tagged these antibodies with the carbon dot, and then when the antigen antibody complex they form, and then they got binded with the specific antibody on the test line. And if you are shining say ultraviolet light, depending on the specific antigen antibody binding, there will be specific emission range spectra. To that extent it is good, but this is happening at such a small scale. First of all, you have to visualize, and then you have to interpret. So to visualize that, you need to magnify, and then you require microscope. So microscope is thought to be a bread and butter of most of the life science related labs. But if we require a microscope, again and you have to bring this back to the lab, then the whole purpose of bringing the test to the ground has to go backward. So what we try to do is very simple. Like, this is almost like a school project that you get some lens, objective eyepieces, etc., and make an assembly of that in the form of a microscope in a 3D printed enclosure. I consider this to be a very good school project and even a first year college project also, because people will get a hands-on idea of what the technology is in terms of practical instrumentation. And this could easily give 1000x magnification. But then after it gets magnified, where do you visualize? There is no question of any spectrometer here. So we use the simple smartphone. So this 3D printed device had a smartphone attachment, and the smartphone will take that outline. And it would be typically a bit blurred and not as precise as you get in any system. But what we used to do is that we already had a calibrated sample, pre-calibrated, which has a known concentration. And then with the image that we get from the smartphone, we use deep learning to extract the features from the image and then correlate that concentration with the deep learned images. So in that way, that AI coming at the last stage, it gets decisive in terms of augmenting the accuracy of such things in a way which I would say even 2-3 years back was not so easily possible. And these are some examples where a physical technology in a simplified manner with AI combined is actually creating a lot of impact in bringing high-end things for the common people. I would also like to give one example on infectious diseases because so far we have not covered that. So during the COVID time, we somehow tried to intervene in the problem of the scarcity of resources of RT-PCR tests. And while during the early stages or the rather early period of the COVID pandemic, lots of media hypes were there about COVID PCR kits being developed. According to me, it was like a much more involved problem and not just develop a kit. A kit will work only if you have a machine. And that having a real-time PCR machine at every remote lab is unrealistic because you need not just one-time cost, but also you need trained human resources and other lab conditions for the maintenance. So what we did is we developed a simplified machine which can actually do automated but piecewise isothermal test instead of cyclic heating and cooling, which the PCR will do. And then we adopted that test, which is actually known as a common protocol in the life science world, LAMP, loop mediated isothermal amplification. So we tried to adapt that in this particular machine. I was working with Professor Arindam Mondol from our bioscience biotechnology. And in that way, we developed a simplified ISO PCR, and it was quite satisfying, of course, because we validated it in ICMR NICET. At that time, the name was NICET. So, and it was all very, very positively going on. But again, although it was commercialized and all, after COVID, this is one of the questions that technology and its development would confront. That after COVID, what is the importance of a COVID test? I mean, after the COVID era, I mean, people might still have COVID now, but it has virtually no impact as compared to what it was having during the second wave of the COVID. Now, because we were working on these technologies with ICMR Center of Excellence, so we got an advisory from ICMR that why don't we adapt this for TB detection. And in fact, we have with NIRT Chennai, we have made significant advancement on this for TB detection in terms of that yes no type of test we have successfully established. Now the second level is something which is also we have advanced well, but we need some more work, and now the field work, the clinical validation, it has started and gained momentum, because TB has this multiple drug resistance problem. Typically, the rifampicin and isoniazid, the two main drugs, and this, if drug resistance develops and if it has a potential to develop, that also should come under the ambit of the detection, and that that work has advanced well. Now, on the other side, I was every time when we do something, as I told, that some incompleteness appears. So all of you, all of us have gone through the COVID test at least once in our that time. So one of the typical things which would make the test successful is the correct collection of the swab. So typically the most accurate is that nasopharyngeal or oropharyngeal swab. It's not just simple nasal or oral swab. Now, when that swab is collected, there is actually a very stringent protocol of how to collect it. And if that collection is not proper, then the test is not going to work. I have seen an example when we were traveling just towards the end of the COVID period. Me and my wife, we were vaccinated, so nobody was asking a question at that time. Even in some of the airports, they were asking for the certificates. My son was much young, so he had no chance of vaccine. So there it was told that you must do a COVID testing with negative answer before he can board the flight. So with the advisory of the our local hosts, we were there in a university system. So university office was fantastic. They sent us to a hospital, and that hospital just simply asked that for travel. Say yes. So they brought then my son to a room. And the way in which his swab was collected, it can never be positive. So, but ICMR certified this sensitivity, this specificity, all these things. But it is like in our young days, sometimes for urine test we put water, in the job medical test. So that kind of thing, if it is done, then the whole purpose is lost. So collecting a swab is actually a challenge. And these kind of tests are very important when infectious disease come in a sudden blast. For example, seasonal flu. It creates a lot of challenge in the junior schools. And in a class, if a child is affected, almost like all, because they mix very intensely and they do not have that control in mixing. So we developed a saline gargle based test, particularly for the young kids. And we developed a protocol for this. We did a first round of validation with the COVID samples which are available. Another ICMR lab, which is ICMR regional lab, RMRC Bhubaneswar. And believe me or not, if we make certain safeguards, this doesn't give that inaccurate result. So, the challenge is that many times people are not careful and they are just giving any gargle sample with not the precaution that is being provided in the beginning, and then there will be some problem. So I always wished that if such simple technologies were given to pediatricians, a child specialist sitting at, they could in their offices do a simple test and make certain screening, which actually is missing currently. Now, again, like the tests that I have mentioned so far, be for infectious or non-infectious disease, they require one common thing. It's a device. Remember the first black box that I showed. So why such a device is required? Such a device is required because you are not having a lab control. So whatever control in terms of optics, electronics, etc., you at least have an enclosure where you can put it. And if you do not keep your device, let us say, someone, in fact, this thing first came to me as a important question when one of my students, and typically, the students who are in junior classes, they may ask sort of crazy but very interesting questions. So I was just, he came to me for a project and I was just talking to him and I was explaining some of the technologies that I wanted. He said that, sir, why do you require a device? So I gave him this kind of explanation. He said, but all the technology needs is a paper strip in which the test is going on and taking a picture. So if I take the picture in my smartphone, what is the problem? So then I explained him that that is where the challenge actually lies. Every smartphone has different camera setting. The background is different, and the human who is taking the photo is also is expected to deviate a lot from any sort of protocol. That means that if you are taking a photo, it becomes an acceptable photo if you are analyzing by image, but it cannot be referenced with any calibration. Your standard calibration will fail and therefore your accuracy can go to any dimension or lack of accuracy. So then interestingly around that time, startup company Smart QR Diagnostics Technologies, they are a startup company from Pune and they got incubated at IIT Kharagpur and they were conversing with me to transfer some of these technologies in a device-free format. And it created a lot of interest in me that at least they wanted to make a business out of this. And the reason of making a business is quite clear, that we have seen many people using the glucometer or the Sakuchek or whatever. So we cannot imagine a glucometer without a meter. And the way in which it puts the challenge is that usually there are many people who are routinely testing their, say for example, blood sugar or insulin or other forms of food and other lifestyle monitoring, but they are otherwise very fit and they are working. So suddenly a meeting is going on, someone brings out from the bag that glucometer, starts doing it, or in the airport or railway station, a busy center. This is so odd. But it could be quite easy if you are bringing out just a paper strip from your pocket. You have a lancet pen in your pocket, so you just prick it, take your smartphone and take the image. And then the challenge is that you are challenged by that calibration puzzle. So what we did is, we tried to apply two things. And this is where I always, for the next generation students, I want to highlight that if you are working on a physical system and if you are working on AI, very nice, but never succumb to black box AI. Try to infuse physics or whatever, chemistry, biology, and fundamental domain science with the AI. So when the light interacts with the paper, there is a light matter interaction physics. And that light matter interaction physics, the essence of that can be incorporated in a relative framework. That means, say there is a paper and light is interacting on it. Now wherever there is a test spot and wherever that reaction is not happening, still the same paper, still the same light matter interaction. So that physics you can use as a reference, and then any sort of signal which comes out of that is an aberration, which will distinguish between different outcomes of the test. And to isolate that signal from the fundamental light matter interaction for that strip, we used, of course, some kind of feature extraction and machine learning techniques. So in that way, we were focusing on women's health. So two important tests we developed. One is HemoQR, which is a hemoglobin test. So by this finger prick, using your smartphone and the mobile app, which can be downloaded from the Play Store, you can quickly get the hemoglobin of your blood. Prepap QR is another test, which is a very unique test for women to screen from their different diseases which could originate from vaginal infection and several other related things. And one of the premises of this is a 1 decimal accurate vaginal pH value, which is not so easy to get, because again qualitative pH paper will not give that. So and one of the major what I would say punch points of this technology is that for using this technology, the woman doesn't have to go to anyone else. Just she has to look into the video and self-collect the swab, put it on the strip, take the photo and get the result. So a huge crisis of women's very special health conditions which cannot be addressed in a primary center or a common center simply because you do not have reliable people or experts there. This is a way in which their health conditions could be monitored in a democratized manner, and they can sort of communicate that particular result with any doctor with whom they have faith or confidence. So I will quickly run maybe one or two of these videos.
HemoQR. HemoQR presents
both are similar, so I will just run one just to give you an idea of how the test works.
Microfluidic reagent. This cutting-edge product combines advanced microfluidic technology with colorimetric analysis for accurate and convenient hemoglobin level assessment. The paper strip is embedded with specialized reagents that trigger color changes in response to hemoglobin presence. Leveraging the capabilities of a smartphone camera, the accompanying app interprets the colorimetric changes on the strip, translating them into quantitative results. This seamless integration of paper-based microfluidics and digital analysis delivers a user-friendly, on-the-go solution for hemoglobin level quantification. HemoQR's versatility makes it valuable for both medical professionals and individuals, enabling quick and reliable hemoglobin measurements. With its portable and efficient design, HemoQR simplifies hemoglobin detection, enhancing healthcare accessibility, diagnostics, and screening. To perform the test, prick the finger using a lancet. Use fresh lancet every time while performing a test. Depending on how deep the lancet goes into your skin, you can set it according to the rotator or in the lancet device. For thin skin, set the rotator at 1 to 2, and for normal skin, set it to 3 to 4. Hold the drop of blood against the strip's edge. Touch the hemo strip on the drop of blood. Scan the strip using the HemoQR mobile application. Get your hemoglobin results within a minute in the mobile application.
So these are the types of tests, and the interesting aspect of the test is more simple it looks, actually more complex the technology has to be. And this is where I find that there is a lot of joy and of course a lot of challenge in working with this. And if I can mention my own experience in working with this technology, the first time when I worked in the lab on this, I never thought that this technology will come to the market and sell. But once it has happened, and currently it is there in the market, what I realize is that there is always a way to come to that stage if you question your output at every stage and do not get immensely satisfied with what comes out of it.
HemoQR.
Sorry. Now, a final example that I would like to give before drawing the final remarks is that so far I have concentrated on diagnostic tests. But we have to realize that there is a significant distinction between diagnosis and diagnostic test. I can at least remember quite well that in my childhood, my father used to take me to a so-called child specialist family physician every time. And one or two such doctors were there. And later on, I realized that in many of those cases, just by asking some of the symptoms, making some essential physical examination, the doctor will come to a recommendation which used to work without a test. And of course, that is why we are all living. Otherwise, we would have not lived in that way. So that was not an era of diagnostic test. But how that family physician could manage it so well is that inherently in the mind there would be a data about the natural, what is says, constitution of the patient, and that would immensely help. So the doctor would know about it even if he or she doesn't look into the previous prescriptions, more or less, because a human has many such intuitions. Even much before that, we have not seen that era, but I have heard from some of our seniors that legendary Dr. Bidhan Chandra Roy, sometimes by staring at the eye of the patient, would say that what the disease the person has. It might appear to be a bit, someone would always have that special ability, and combining that with, this storytelling, sometimes it becomes like a myth. But if you decode the reality, there is always a possibility that someone will have a better intuition of extracting some features. Like a very good doctor, even nowadays, would carefully see how the patient is entering into the office, sitting, and that also adds to the interpretation in a very subtle way that will not come in a computer algorithm. So diagnosis is not just about diagnostic test. There are many features by combining the medical history, symptom, and outcome of an essential simple physical examination. Many features could be arrived at which would, if not give a confirmatory test, at least can give a good screening test. Now, what a good doctor does is that typically one or two symptoms could be due to many possibilities. Like someone is having a headache. If you write to Google, Dr. Google will start with possibly brain cancer, because that's the most impactful thing, most times downloaded page, so that will come at the top. And by that thought only that person will faint. So, but it could also be due to simple migraine. So, a good doctor will try to look into many other things and eliminate possibilities, and maybe from 1000 possible reasons of headache will come down to 5. And then to resolve the dilemma of those 5 might require first a screening test and then some confirmatory test. That is what most good doctors will do. Sometimes no test is also required. So a few years back, just before the COVID pandemic, we developed a very unique technology for oral cancer screening, and it is something like this. We worked in collaboration with the Guru Nanak Institute of Dental Sciences and Research. So the camera, of course, now the camera looks very different. We are commercializing it in a different way. This was a testing stage where the clinical trials were being done in the hospital. So this is a thermal camera. So it takes the pixelated temperature data from a lesion which is there somewhere inside the mouth, and by our algorithm it converts the temperature image to a blood perfusion. That locally in the micro scale, how blood is perfusing. And most of the cancer related progression, typically in oral cancer, you have oral submucosal fibrosis, which is known as pre-cancer. And then at some point of time, it might get transformed into oral squamous cell carcinoma. Biologically, it is epithelial cell converting to mesenchymal cell. And when these things are occurring, you will see a change, dramatic change in the vascularity, and therefore the local blood perfusion will change. So this technology we developed the stage 1, the field trial was quite good. We published this in the PNAS USA. But then the question came, of course, many of the doctors who handle these kind of patients, they said that, can you give us a technology where we can use it more holistically? because most of the patients who are coming, say for dental or oral purposes are not related to oral cancer per se. They have many other different problems. So then we found that it has its own advantage but also own limitation. So what we did is we developed a simple app where you have inputs which are being given by the patient or patient's friend, whatever, some standard images and basically a comparison of that with what is being visible, and also this chewing habit, etc. And then when this is combined with this technology, individually they may not be decisive, but combinedly they can actually give a good clinical decision support by optimizing the weightage of each of the inputs, which will come from real life patient data. So I think this is where a significant amount of clinical decision is going to be there. Well, of course, a human doctor will look into it and validate it. If the job is not that automated technology will take this, but a doctor who throughout the day is addressing, say, 500 patients, for that doctor, it will give a set of quantitative precise inputs to look into, and then it helps the clinician's life in a much more simple way. So in this way, what we have experienced so far is I would say, at least to my understanding, a very transformative journey. From my own student perspective, when I considered lab as the final thing, I would realize that lab is the starting point of these things. And now I realize that the kinds of mistakes that I did in my early days of working in these research topics, if I had gone back to those days, I would not make the mistake. The first mistake is designing the thing in the lab for lab work, never keeping in mind that when it goes to outside those controlled lab settings, then whether it will work or not. And when we test it in the field and find that it is not working, almost a re-engineering we have to do. Many cases I have done that. Right now, all the technology that I am developing, of course, I am doing it from a different perspective. But in my early days, I made a lot of mistake on that. So, but mistake will give you experience and insight. And in this way, what we found is that it is not just like a technology. You can connect it with people, rural guards, who are trained with these kinds of technologies. Before I became the director, I did not have an opportunity to introduce these kind of courses in the IIT curriculum. But we used to train them just like outreach courses. Now IIT Kharagpur itself started a allied skill development program, and we are giving government certificate for these rural boys or girls who are learning the allied health worker along with these technologies. And the group of Dr. Sotodal Saha from the Foundation for Innovations in Health, and being himself qualified FRCS doctor, he is immersed into these kinds of activities, and I have found that it has been a very enriching experience, from our side.
Professor Chakraborty's groundbreaking.
So what I would imagine is that at the end we are trying to emphasize on, of course, science, and this is sometimes a misunderstanding on people, that when we talk about great instruments, very sophisticated things, people will say, that is science. Now when something which works so easily and apparently like almost instantly gives something and it works, in India people will often call it jugaad technology. This is an Indian name. Now what you have to realize is that a jugaad technology cannot survive if it doesn't have the backbone of science, because it is very important to start from the foundations of science, and not all foundations of science will lead to this, but whatever leads to the final outcome, if it is connected with the foundation of science, actually you can develop in a systematic step-by-step manner. So while we are in an era where AI augments human cognition, my learning from all these is that the future belongs to those scientists who can design experiments informed by data science, but do not rely only on AI. Build models that can integrate physics with machine learning or AI, and bridge molecular scale understanding with the large scale consequences. And not just the the medical technology, but climate or other forms of sciences and technology will require this. So just as my final remark, I was a bit hesitant in presenting this as a material of the talk, given that this is really a pioneering institute of basic sciences. But and I am no good in any of the basic sciences up to class 12. Whatever I studied, I studied. After that, it's all how engineers study physics, maths, etc. All the real physicists and mathematicians will laugh. That's one of the hybrid ways that we somehow manage our engineering work. But what I would say that at least these kinds of institutes are very important in the modern perspective, because science at the first level should be driven by curiosity. This is my own personal feeling. It should not be immediately driven by commercialization. On that fundamental level, that curiosity-driven science and impact-oriented science, these appear to be a bit opposite, but they are not, because I believe that they are phases of the same continuum. We always start with a curiosity-driven science, and for example, when Sir Raman explored light scattering, he was not thinking of how the Raman effect is used for optical diagnostics. Nowadays, yes. The mission of basic science research institutes, therefore, at this critical juncture, there is always a tendency to dilute fundamental science in the name of utility. It should not be. Fundamental science should be given its fundamental importance, and that can feed the supply line of so-called technology development. So I believe that institutes like these are great, and they should continue to produce scientists who not only study nature but try to decode its complexity, not just publish knowledge but expand human capability, not of course merely work within labs but transform the living world beyond them. That vision has to be there, but that childlike curiosity, if it is missing, I think that the entire spirit of science is lost. So my 3-point summary to some of the students or the young researchers, if they are present: dare to ask unfashionable questions, build bridges across scales and disciplines, and measure success not only by citations but by transformation. Thank you very much.
Thank you, Professor Chakraborty, for giving such a fantastic overview. And as you said, we are a basic science institute, but you very nicely connected it to, really to the life, from lab to life. So we can take a few pressing questions. Yes, please.
Fascinating talk. One of my concerns is the false positive that we get when we use technology, which is not as sensitive as what is being used in the clinical settings. Probably with your AI, that gap is closed.
So this is a very important question. In fact, in just to summarize, in any medical diagnostics, we use the two terminologies: sensitivity and specificity. So sensitivity is true positive being interpreted as positive, and specificity is that true negative should be interpreted as negative. And that is of more concern. Even in the COVID period, I have seen 1 or 2% COVID negative, but somehow COVID positive. Some people almost isolated them in a jail type of place. So what I tell always to my students, the way in which to do it, in fact, is easier to do this kind of elimination of false positive. That any sort of test, when it requires validation with samples, we actually have abundant of negative samples around us. But we are always much concerned that we go to the hospital and take the real, affected patient samples, etc. Sometimes for all these technologies, I tell some of my students, in my lab around 50 people are there, some project staff, student, etc. If each of them donate a blood sample, it will already become a nice cohort of mostly eliminating false positive. So the whole idea is that eliminating false positive is actually more important than detecting true positive. So you are very right, and AI helps a lot in it. But you need a lot of diverse data to actually train the AI for that.
Thank you. Wonderful talk. Wonderful talk and very impressive technology. What I want to ask is that what is the being done to teach this technology across the country to villages, small health clinics where they really need to have cheap and easy testing methods.
So this is a very, sorry, your question is very important. What we normally do is that this is where we use government support to the maximum, because all these, at least 90% of the technologies that I have shown are connected with two important government-linked projects. One is a common research and technology development hub, CRTDH, on affordable healthcare, which is DSIR supported, and the other is ICMR COE. And typically, the DSIR supported part helps in the manufacturing and the technical component, MSME, mobilization, etc. The ICMR part is helping a lot in getting into the regulatory and the trials and implementation. They have some mission mode national programs, and in fact, very recently, that hemoglobin test, it passed that. So as per ICMR advisory, any health center in the country can use it. So that very recently we got that certificate.
So if there are no further question, I may want to ask. So we are a basic science institute, but we also have a technology research center. And one of the challenge as you go from lab to life is the valley of death. And maybe as an engineer, you had a different approach. But as a basic science person who never had heard of patent in life or something, if they really want to get into such kind of thing, what will be your advice to overcome this valley of death? Because that's really a problem.
It's a very important point. But as engineers, we also face the same valley of death. It's not that we do not face. And like if you see at the end, engineers do not have dramatically different training as compared to the scientists. I mean, never we in our 4-year undergraduate program had a real practical thing except in the engineering labs and workshops, which are simulated versions of the practical thing. So in that way, it is also a very important learning for all of us. But I consider the valley of death as a valley of opportunity, because if you see at the end that you have say taken it to a TRL 3-4, which normally out of the lab prototype, that is the maximum that you can do. Then it is more of a cultural problem. It's if you are careful enough and if you interact with industry or the startups who are working in the domain, you will at least know that what is the gap between what you have developed and what is the need of the market. And if there are supporting people who are helping to work in bridging that gap, maybe in all technology it's not possible, but if 10% of that is there, that's good enough. Remaining 90%, it advances science. It's all good. It's not that it's useless.
Thank you. So maybe with that we close this session, and I think can you please keep it brief? Yeah, please.
So hi, Professor. Hello. So I closely follow your policies and your YouTube videos, also the interviews. So I just wanted to ask that your research outputs are most attractive to us, like the basic researchers, right? So we do basic science, basic studies. So what would be your advice to us that because our research output are not that practical in that sense. So what would be your advice to us?
Don't care about all those. I was just telling likely to someone that we all hear about the Newton story. Newton was sitting and apple fell on his head. He would have usually died if few apples fell on his head with the dynamic load. But if you take that as a spirit, then did Newton realize at that time that Newtonian mechanics will drive so much of technological advancement? So just see, basic science You are gifted with a curiosity-driven approach. Follow that. Do not bother so much about people what they are saying. Stick to that, come to good output, and once you see that output has a practical possibility, make a collaboration with industry or your friends in technology. But you don't have to be jack of all trades. You stick on your basics, stick on your curiosity-driven science.
Thank you, Professor Chakraborty.
Thank you so much. On that note,